Okay, well, thank you very much for that introduction. As you said, my name is Richard Bargett, and I'm the incoming president for the British Ecological Society. It really is an enormous pleasure for me today to introduce our first plenary speaker, who's Professor Sue Hartley from the University of York. And I was thinking last night when we brought this plenary lecture forward how I would introduce Sue, and I was thinking about it, and I would say Sue is quite simply an extraordinary person. I've known Sue since, I think, about the mid-1990s, when we first met at Silwood Park, when we were involved in what was the Ecotron experiment that Heffin Jones at the Frontier was also involved in. And I would say quite simply, I've never met anyone who is as enthusiastic, as committed, and as capable in terms of her research. I was talking to some of her colleagues from York University recently, and they described her as superhuman, and also as the busiest person on earth. And I think, in some truth to those different points. Now, many of you will know about Sue's research, and her work is really focusing on plant-herbivore interactions. And she's worked across a very broad range of systems. She's worked on birch forests, she's worked in moorland systems, she's worked on tropical seedlings in Borneo and other tropical countries, and more recently she's worked in agro-ecosystems that I think she's going to talk about in her lecture today. But I guess at the core of her research is really identifying the chemical mechanisms by which plants defend themselves against herbivore attack. And really this is what I guess she's best known for in her research. And this goes right back to her time when she did a degree, which was at Oxford University in biochemistry, but I guess she saw the light and rapidly realized that the right direction to go was in terms of ecology. And she did a PhD at York University with another past president, John Lawton. Now, ever since I met Sue, I think, as I said, it was in about 1995, I think, when we first met during the Ecotron years, she's been omnipresent within the British Ecological Society. I've actually written down a few of the roles that she's had here. She was a council member, 1995 to 1998. She was vice president, 2004 to 2007. She chaired the publications committee from 2006 to 2011, which was a time when she guided significant change within the publications. And then she's been president of the society for the last 2 years. And she's been recognized through her work for the society and more widely through the President's Medal in 2009 and the BES Board in 2011. Now, if that isn't enough, Sue also has a major role in her own university. She's director of the Environmental Sustainability Institute, and she's chair of numerous advisory committees. And then in her spare time, I guess, she also found time to do the Royal Institution Christmas Lectures, which in 2009, which is an institution for British children. And she was, in fact, the fourth woman to do that since its inception in 1825. So I can sense that she's chomping to get on the stage. So just to sum up, it really is a great honor for me to introduce Sue. It's a great honor for me to take over from Sue in her role as president, which I'm under no doubt is a significant task, and she will be a hard act to follow. She's done a fantastic job in the society, and I think just looking around here at this vast audience and vibrant meetings is really testament to the work that Sue's done. So before she comes onto the stage to do a talk, I've also been said that just to emphasize that questions should be given through Twitter. I've never used Twitter in my life, so it's going to be quite a challenge towards the end, but hopefully Sue will be able to guide me in the questioning. So if people do have questions, please do introduce them through the use of Twitter. So without further ado, I can see she's almost off her chair, so Sue Hartley.
Thank you so much, Richard, for those kind words. You can see why I wanted Richard to follow me as president. Before I start, I've got another thank you, and that is to Wiley for sponsoring this lecture, and indeed for all the work that they do for the Society. Without Wiley's publications and their contribution to the Society, we would not be able to run events like this. So we're extremely grateful to them for all they do. It's going to be a short talk. There we go. We've heard from our excellent introductions about the importance of interdisciplinarity and the importance of collaborations across borders. And I want to emphasize that during my talk. And I want to show you how thinking about classical ecological problems, the interactions between organisms and their environment, can help us in addressing real-world problems, the challenges we've got to face at the moment of food security, climate change, and so on. And what I'd like to show you is that strength within the discipline of ecology, through classical ecological research, can link with other disciplines and collaborate internationally to deliver impact, to address these challenges. So I'm going to start with my classical ecological work, you might say. As Richard very kindly pointed out, I've been working for a long time now on the questions around interactions between plants and their herbivores. And I've always been interested in how plants defend themselves against herbivores, what effect that has on herbivore performance and behavior, and the consequences of those interactions for plant communities. And as Richard mentioned, I've studied those interactions in many places, in the Sinai Desert, in the jungles of Borneo, and in Mediterranean ecosystems. And I've asked these three questions. I might also ask why I only seem able to afford one pair of boots and 2 shirts. What have I learnt from those studies? Well, I've learnt that herbivory is hard. Herbivores have difficult choices to make. Sometimes they just give up and sulk. This sheep's decided that sand is a better option than this extremely unpalatable plant in the Sinai Desert. And herbivores often adopt extreme solutions. I've just been spending some time in Australia, and koalas, of course, have an unusual and very specialized diet, eating only eucalypts. And they have a sort of lifestyle that I aspire to, 4 hours of eating, 20 hours of sleeping. And pandas are another specialized diet, and here I am at a trip to the Great Wall in China as part of the Intercol conference, and I'm wondering about panda diets, but mostly I'm wondering why I spent $20 on a cuddly panda with a piece of plastic bamboo in its mouth. So why are plants so unpalatable? That's been the question that I've been considering. And why are some plants in particular very unpalatable? And what was striking when I worked on heather moorlands was under heavy grazing pressure, you can see that the heather is in severe decline outside the fences, doing okay when you fence it off. But outside the fences, particular grass species thrive. And one of those was Nardus stricta, or markgrass. And I'd always been interested in chemical defenses, in those toxic chemicals that helped plants defend themselves. But it was a colleague from Manchester, Roland Enos, who's now at Hull, actually, and he said, but actually, Sue, the important thing about Nardus is physical defenses. And he pointed out that Nardus, like many grasses, takes up a lot of silicon. And that silicon is deposited in very abrasive and difficult for herbivores to tackle structures. So I thought it would be interesting to study silicon, and so it's proved. So just a little bit of background to silicon defenses. Silicon is a very abundant element in soil. It's not always easy for plants to access. That does depend on environmental conditions, but many plants are so-called hyperaccumulators of silicon. They're extremely effective at uptaking silicon from the soil, and grasses in particular are high accumulators, and they can accumulate up to 10% dry weight of silicon. So that's more than many essential elements. And so it's taken up as salicylic acid in soluble form through mechanisms that I'll talk about later, deposited, and then once it's deposited in these very solid forms, so it's very hard for the plants to remobilize that. So once it's deposited, it tends to stay in the plant until it's either the plant's eaten or senesces. And these structures make plants really quite hard for herbivores to deal with. The sort of effect of eating those is very well illustrated by this slide. It really is like those nightmare scenarios when you went to the beach as a kid, your mum had made you sandwiches, and the sand got in. Nightmare. So it's very abrasive and difficult for herbivores to deal with. So early experiments looked at how insect herbivores coped with that kind of abrasion. So in these graphs, we've got the consumption of a range of different grass species, Poa, Lolium, Festuca, and Brachypodium. And we've got black bars here are low silica plants, and white bars are high silica plants. And you can see that these armyworms and these locusts consume far more material in these preference tests from low silica containing plants. So silica is having an influence on the herbivore preferences. When we looked at grain aphids, they seemed far less concerned about silicon, and really silicon did not have much of a negative effect on the abundance of aphids. More on why that might be, it took us another 10 years to understand that. So silicon also reduces herbivore performance. So this is looking at more grass species, Dysampsia, Festuca, and Lolium again. And this time, just keep you on your toes, the black bars are the high silicon plants. This is the relative growth rate of the insects. And you can see that in all cases, the growth rate of the insects is lower on the high silica plants. And that's reflected in the amount of nitrogen that the herbivores are able to absorb from the food. Now, herbivores are often nitrogen limited. Plants are relatively low in nitrogen compared to herbivores. Why might it be more difficult for herbivores to absorb nitrogen from foliage rich in silicon? Well, it relates to that abrasion that I was telling you about. The great thing about working on insect pest species is that nobody minds if you chop their heads off and look at their jaws under the microscope. And this is the mandibles of an insect reared on a low silica plant, and you can see there's quite amount of tooth here, if you like, whereas when the insects have been reared on high silicon plants, you see that the mandibles are very much worn, and we can quantify that and see that on high silica plants there's a greater degree of wear. So that's the situation with insects. I want to think about larger herbivores. Now, we don't know anything like as much about the influence of silica on mammalian herbivores, particularly large mammalian herbivores. There's obviously something funny happens to herbivores when they eat grass. This sheep seems to have developed two heads. But we don't actually know much about the influence of silicon on sheep. But we have done one study which involved videoing lots of sheep. So if you, I mean, I might be sending you to sleep now, but certainly if you are struggling with insomnia, sheep videos can really help. So this is looking at the bite rate on Agrostis, Brachypodium, Festuca, Lolium, and Poa, and silica, with the exception of Poa, seems to have a bit of an impact on the bite rate. And that's important for ruminants because they need to maintain an intake rate to keep the rumen functioning and happy. And there is also some evidence that silicon has adverse effects on rumen microbes. There are some groups that we know absolutely nothing about. Nobody has looked at the effects of silicon on marsupials. As Jane de Gabriel, a colleague from Australia, and myself discovered when we did a review of this recently, I think catching them to actually test it might be quite a problem. But there is one group of mammals that we do know quite a lot about, and that is voles. Now, Xavier Lambin and his group in Aberdeen, together with colleagues in Liverpool, have been studying the population dynamics of voles in Kielder Forest. And like many small mammals, the voles show these distinct cycles. And that's a problem that's been a sort of classical ecological issue for a long time, and ecologists have been very interested in trying to understand what drives those cyclic changes in population. And the tradition has been that this was a top-down effect with diseases or predators, and that as prey populations increased, predator populations tracked that and eventually brought prey populations down, so predator populations also declined. But there is, of course, another possibility that we were interested in testing, and that is whether changes in the quality of food through silicon could also have an impact. Now, to create cycles, we really need causal factors that are density dependent but show a time lag, and that show a dynamic feedback between the herbivore density and the plant functional response. So can silicon fulfill those criteria? Well, when we looked at the effect of silicon on voles, this is for Stucrovina and Lolium again, high silica plants and voles certainly prefer to avoid the high silica plants and eat low silica plants. The same issue they suffered as the insects, that when they fed on high silicon plants, they couldn't access as much nitrogen, and that had quite an impact on their growth rate. In fact, they showed negative growth when fed high silicon plants. So there's certainly an impact on growth rate, and in voles, it's body weight that determines when voles become reproductively active, and so there was a potential mechanism there. But what about the delay factor and the density dependence? So we looked then in a lab study at the way that silicon defenses changed under vole grazing. And we noticed two things. So there was a period of treatment here, of damage treatment, a low damage treatment of only 5%, and a high damage treatment where the voles removed 20% of the foliage. And then we stopped the damage and observed what happened to the silicon levels. And two things strike you about this. First of all, that the damage level needs to be above a certain threshold before you see an increase in silicon defenses. So the density of voles is critical, because if it's not high enough, it doesn't produce damage levels that are sufficient to induce silicon defenses. And the second thing you'll notice is that silicon defenses take quite a few months to accumulate to their maximum level, and then they take quite a long time to decline. So we have this kind of delay between a high density of voles and the silicon levels. So far so good, but that's the joy of lab and theoretical work, isn't it? When you get out into the field, the situation changes. But so far so good for our hypothesis. So we have high vole population density, elevated silicon in the grass, reduced vole growth rates, low voles population density, reduced silicon, and increased growth rates. It was all going so swimmingly well. And then we went out into the field, where it didn't go quite so well. There seems to be something about snow in my life. So the field conditions were extremely difficult. But the field team, I was notable by my absence, of course, but battled on manfully and indeed womanfully. And we did manage to set up an experiment where we had enclosures with different densities of voles. And the idea then was to see if vole density affected silicon induction in the field. And we also performed a very large-scale experiment where we trapped and removed voles from whole hillsides at Kielder. So quite a superhuman effort. So what did we learn? Well, we learned that ecologists sometimes come to resemble their study organisms, a certain sort of volish characteristics. We also learned that voles don't particularly like being in enclosures, and they would kind of scramble up here, clamber out along there, then discover they couldn't go any further, drop off, and try again. So hours of endless entertainment. But we also discovered that in the field, things were very, very different. And if the reviewers see the light, you can read about the results of that study in Ecology and Evolution. But I will just summarize them to say that we found that we could see induction of silicon defenses at even the landscape scale over whole hillsides. But those silicon levels did not reach sufficiently high levels to influence vole growth rates. And the reason for that was that the damage levels in the field are generally too low. So where does that leave our hypothesis? Well, we can certainly see some induction of silicon defenses, but not to the degree or to the long-lasting, or for long enough to actually increase, to affect the vole growth rate. So we haven't seen the evidence for this hypothesis at the field scale, but nothing ventured and all that. But of course, the reason that things are difficult to demonstrate in the field is that we're dealing with variation. And we were dealing with big variation in damage levels and in induction levels. And that led on to a different way of thinking about silicon defenses. And I want to think now for some of the rest of the talk about what drives that variation and how we can use it. So I hope I've shown you that silicon is an effective defense, that it can deter feeding, that it reduces growth, and that's probably because it's very abrasive. So when we measure abrasiveness here by looking at how grasses can cut grooves into perspex plates and correlate that with silicon, we do see quite a good relationship. So generally, the more silicon a plant has, the more abrasive it is. But that isn't always true. And here we've got two plant species, Festuca ovina and Festuca rubra, that are quite closely related in the same genus. They have similar silicon levels, but wildly different abrasion levels. And we can see that again here when we grow Festuca on a low silicon medium, so it has very low silicon levels, it produces just as much mandible wear as Lolium with much higher silicon levels. So there's clearly not just how much silicon you have, but what plants do with it. The other thing I think it's important to note is that silicon is an inducible defense. I've already shown you need sustained damage to increase silicon levels. That was demonstrated again in another study in the lab looking at control plants, mechanically damaged plants, locust-grazed plants, and vole-grazed plants. And a single damage event does not cause induction. Repeated damage is required. And we also see that, excuse me, clipping produces rather less of a response than real herbivory. And that isn't unusual. Plants can detect when they're being attacked by real herbivores compared to a pair of scissors. But what was also interesting in this is that when we compared different genotypes of species, we saw very different effects. So here are two species, Deschampsia cespitosa, which I've mentioned before. It's very high in silicon. When it's clipped, it tends to respond with an increase in silicon levels, but not always. Some genotypes are much less responsive. And this is a plant called Avenella, and that shows very little response. But again, sometimes genotypes do show a response. So there's big between species and within species differences in induction. Why is that? Well, to understand that, of course, we have to know more about how silicon gets into plants. And this is the first illustration, really, of the importance of working with other disciplines, with plant geneticists and with plant physiologists, to understand more about this process if we're going to get to grips with the ecological consequences of silicon uptake. Now, silicon uptake is quite complicated. It relies critically on a transporter called LSI1, which is an aquaporin-type transporter. It's a passive transporter. It just allows water and other things to flow into the plant roots. To get out of the root and into the xylem is an active process. It requires an efflux transporter, a proton pump, and that's an energy-dependent process. Then there's transporters whose job is to sort of distribute the silicon around the plant. LSI6 is involved in that, and a more recent discovery, LSI3. Nobody seems to quite know what's happened to LSI4 or 5. They seem to be out of the picture. Once we get to the leaf in question, LSI6 is important in taking it out of the xylem, but how it's deposited within the cells, inside the cells or in the cell walls, we really do not have any idea. And that's a bit of a shame because it's quite important. But what we can do is see the effects of having those transporters not being present. So this is the work of Marr, who first discovered these transporters. And if you knock out LSI1, you see a big drop in yield. You see much more insect damage. So rice plants that have no LSI1, and in rice, this was the transporter was first identified in rice, then it's very bad news for the plant. We also know how to measure silicon quickly and easily. It's not an easy substance to measure, actually. Chemically, it's very recalcitrant. To measure it accurately needs lots of hydrofluoric acid and lots of health and safety assessments. So it's much better to do it through portable XRF. My group sort of pioneered how that could be done effectively in plant material. And you can also, so that not only allows you to rapidly assess silicon across a great number of samples, but it also means that you can link that to a scanning EM and actually see where the silicon is deposited. So you can see silicon here on this barley leaf associated with these spines, within cells, near the cell surface, and in the tip of spines, and in the cell beneath it. And you can even watch induction take place. This is an undamaged plant with silicon quite localized, a damaged plant with silicon running right across the surface. So with these techniques, we can see what plants do with their silicon. So we returned to the Festuca rubra, Festuca ovina, and Deschampsia question, and looked at how silicon was deposited in those plants. This is Deschampsia, and you can see there's no added silicon here or no damage, and you can see the silicon is in these fearsome spines. This is a very unpalatable plant. Festuca rubra, the silicon is all over the surface, and there are relatively few abrasive protrusions. And Festuca ovina looks like it's got measles. Silicon very localized in these protrusions. When we look at how the plants respond to silicon addition or damage, we see that in Deschampsia, you see a much bigger deposition of silicon and some additional protrusions, many more spines form. The same with Festuca ovina, lays silicon down much more thickly and produces new spines. Festuca rubra doesn't really do very much. So that sort of illustrates why we see such a big difference between the abrasiveness of Festuca ovina and Festuca rubra with the same amount of silicon. It's down to the way they use the silicon they acquire. And the strategy of Festuca ovina is to put it into abrasive structures. And Festuca rubra does not do that. And you might think this is a silly strategy in terms of insect herbivory, maybe. But it might be a very sensible strategy in terms of pathogen resistance and making the surface very impenetrable. So plants have different strategies. They have different approaches to how they use their silicon. And we wanted to study that in more detail and get to grips with why that was happening. So this was a project with Emma McLaren, a PhD student, sponsored by DLF Trifolium. And they're grass breeders. And they worked on tall fescue. and they used to grow different varieties of tall fescue for different purposes, for forage grasses or for turf or for sports fields or what have you. And they classified these grasses by growing them in the greenhouse, wandering up and down, and running their fingers through the leaves and saying, ooh, a harsh variety. Ooh, a soft variety. A very, very soft variety. Semi-harsh variety. We thought that wasn't quite as quantitative as it could be. So we decided to take a look at these varieties. So this is a harsh variety. You can see lots of spines. This is a soft variety, far fewer. And when Emma looked at the silicon uptake across these varieties, you see, I'm really not joking. Very, very soft, soft, semi-soft, semi-harsh, and harsh. But when you look at the way that silicon is taken up, you can see there's a good relationship between the harshness of the leaf surface and how much silicon is taken up per gram of root material. So then we wanted to see how those plants responded to damage, and that proved quite interesting. So this is very, very soft, harsh. I can hardly say this without laughing. Undamaged plants, undamaged leaves in damaged plants, and damaged leaves in damaged plants. So you can see that very, very soft doesn't respond to damage very much. With the very soft variety, you get some induction, but only in damaged leaves and damaged plants. And the harsh variety has a much bigger response to damage. It actually takes up far more silicon when it's damaged and deposits that in both damaged leaves and undamaged leaves. So there's a sort of systemic response. Now, when we looked at transpiration rates in those plants, we found that these patterns match pretty well transpiration rates and stomatal density. So the reason that harsh plants take up a lot more is they transpire much faster and they have more stomata. So they're able to slurp up the silicon, technical term, from the soil more effectively. So we thought, well, this is all about passive transport. It's just about what comes in through LSI1. So we thought, we'll have a test of that. We're nothing but sophisticated at York, so we just thought we'd just shove them in a bag. If we put the plants in a bag, then the humidity is high, transpiration is changed. What then happens? And we got a complete surprise. Well, one thing wasn't a surprise, and that is that without those transpiration differences, the differences between varieties in silicon levels seem to disappear. So the basic transpiration sets the kind of baseline silicon level, if you like. But what we found was that all genotypes, including the very, very soft, managed to show an induction response. So we knew that the induction response was not due to transpiration. This is an active remobilization of silicon in response to damage in all three genotypes. Why might that be? We've got to look at the genetic controls over silicon uptake, and they are very precise. So this is data from a colleague of mine at York, Franz Matthias, and he has a number of wheat genotypes. He took 15 of them. He grew them in hydroponic solutions with exactly the same amount of silicon, 0.5 millimolar. And then he looked at how much they took up. And it varied dramatically. And these are plants growing in exactly the same conditions, exactly the same access to silica, very, very different uptake. Now, when Ma did that with rice, he found in two varieties of rice, Nippon-Bare and Klasov, that Nippon-Bare took up far more silicon, and it had a higher expression of LSI1 and a greater density of LSI1 transporters. So Emma's thought was maybe these genotype differences in tall fescue are due to different expression of the transporters. So she took RNA from the roots of damaged and undamaged plants for the harsh and very, very soft varieties, and she did this stuff. I'm an expert. qPCR. And she determined, this is the joy of working with other disciplines, you just need to be expert in your own discipline. And she did this analysis of Trinity pipelines and EdgeR. It all sounds very exciting. Anyway, what did it show? I'm a great supervisor to my students. That's a good thing about students. They educate you. It's wonderful. So in undamaged plants, when she compared VVS and harsh, there were no differences, in fact, in NSI1 one expression. She didn't actually see that, but she did see that there was an increased expression of LSI2. And if you remember, that's the efflux transporter. So that's the transporter that takes things out of the roots, gets them into the xylem. And that was expressed more strongly in harsh than very, very soft. And there was a similar result in the damaged plants, that LSI2 is upregulated in response to damage, but only in the harsh variety. So it may be that these are sort of fairly preliminary results, but the indications seem to be from Emma's study that LSI2 was quite important in these differences. Okay, so what do we know about silicon uptake in tall fescue? What affects it? We know that transpiration does, we know that damage does, we know that silicon supply does, we know that genotype does, we know that silicon transporters do. And we also now know through other work that defense genes do. So silicon has been shown to be important in priming other defense mechanisms in the plant, chemical defense mechanisms dependent on the jasmonate pathway. So you can see that silicon uptake is actually a very, very complicated process. And just going back for a moment to think about our differences between insect feeding guilds, you can see that these complex patterns of deposition could explain some of the results that we found earlier. So this is work from a colleague of mine in Australia, Scott Johnson, and other colleagues, and he's produced this wonderful graphic of why we think that effects on chewing herbivores are more pronounced than on aphids, because of course surface deposition will have a much bigger impact on chewers than on phloem feeders, and similarly within cell deposition, because of course the aphids can avoid this sort of effect of silicon on their way through to the phloem. And it's only really when the silicon is in the cell wall that aphids may be affected. And similarly, the jasmonate pathway usually has a bigger impact on chewers than aphids. And Scott's performed a meta-analysis recently on the effect of silicon supplementation on different herbivore guilds, and he's found that in general, the phloem feeders are less affected. So we have mechanisms now to explain our ecological observations. And why is this all beneficial? What is the good news about silicon? Well, of course, we've got issues with pests. We currently have a very dangerous reliance on on synthetic chemicals to deal with pests. So this is rather frightening statistics here from, or numbers, about the losses of our major crops to pests. And if we have no pest control, we lose between 1/2 and 3/4 of the yield. Even with our best efforts, we're losing around 1/3 of our food production to pests. And that's not sustainable. We need that food. If we can decrease losses to pests by 1%, we can feed 25 million people. And the other problem we've got is that a lot of these compounds that we use in current methods of crop protection are harmful and being withdrawn. So we have lost a lot of those chemicals. So we need to replace them with more natural solutions. And one thing we've done is bred out a lot of our crop defenses. These are wild potatoes. You perhaps not even recognize them with the colors and shapes, and a lot of that color is due to toxic chemicals. We've bred those out because we like these potatoes a lot better. And we make up that difference by using synthetic chemicals. But of course, there is some good news. Most of our key crops are grasses. Okay, we've modified them hugely. This is what we've done to maize, and wheat is a triffid, frankly. But we've done all sorts of things, but they're grasses. So do they use silicon defenses? That's the hope. So there's been a lot of interest, silicon in agriculture, benefits of plant silicon for crops. I love this. Establishing broad spectrum disease resistance in plants, silicon leads the way. And that's from someone at Ghent University, so it must be true. So, is silicon really a silver bullet, like this paper from Andy Mahag? Can silicon really mitigate biotic and abiotic stress and improve our crop yields. So I've been interested more recently in how we can exploit variation in silicon uptake for practical benefits. I've shown you all the different ways that all the different processes that impact silicon uptake, but can we use that to deliver some improvements in terms of sustainable crop protection, but also resilience to climate change. But the first thing we've got to do, of course, is show that those defences still exist in crops. We may have bred them out. This is just a summary of what I told you earlier from Mayer's excellent paper in New Phytologist. 203 crops, the most common trait change during domestication from wild to early domesticates to modern varieties, and we take out a lot of genetic variation, a lot of traits. So rice, for example, our modern cultivars of rice have 10,000 fewer genes than their wild relatives. That's a lot of traits to lose, and a lot of them are related to defense. So have we lost silicon? Well, this is work by colleagues at Sheffield, Mark Rees and Colin Osborne, and a PhD student, Kimberly Simpson. And they looked, together with myself, at the silicon levels in wild progenitors and in land races and in modern cultivars. And this was across all the comparisons we made, and this is those particular domestications. So here's barley, here's rice, here's maize, here's wheat. And generally we found that the loss in silicon defences was very small, generally less than 10% over the period of domestication. So our crops have maintained silicon. So it's clearly important for a whole range of things in crop plants, and we haven't bred it out. So that's good. So we can use it. We can use it in crop protection, and I've shown you how effective it can be, but can we use it to deal with another threat that we face? And that's climate change. This is a cheery graphic from the FAO about the impact on crop yields by 2050. Red is bad, green is good. So generally, an awful lot of the world is bad, but the places causing the problem is good. There's not a lot of justice in the world. And one of the reasons, of course, that there will be an impact of climate change on crop yields is that we will see an increase in water scarcity. And again, red is bad, and there's lots of red. Oh, look, near normal. The UK is normal. Discuss. So, can silicon help with this? Well, going back to those 15 wheat cultivars, the reason that Franz was keen to look at silicon was that he is interested in the resistance of crops to drought and to salinity stress. And he found when he grew these 15 cultivars that the impact of drought on growth, the negative impact of drought on growth, was much greater in plants that had low silicon levels. And the more silicon plants had, the less that drought stress affected them. And when he looked at why that might be, he found that there was a correlation between the silicon levels in the shoot and the potassium levels in the shoot. And Franz assures me that potassium levels and sodium levels in the shoot are extremely important in combating drought and salinity. And we now have a Newton-funded program with colleagues in the Philippines and in Vietnam actually trying to identify the genes that are responsible for the silicon interactions with potassium and sodium. And if we can identify those genes, then we can cross those into elite cultivars of rice and improve the salinity tolerance. Extremely important in Vietnam, where saltwater inundation in the Mekong is threatening the bread basket, well, the rice basket of Southeast Asia. But I want to actually consider another example where variation in silicon content can be important in addressing a problem relating to climate change and food security. And that is a Global Challenges Research Fund project between York, Dundee, Aberystwyth, Vietnam and the Philippines, looking at improving the use of rice straw. Because, of course, when we grow rice, we only eat a tiny bit of it. So for every ton of rice grain we produce 1.5 tons, at least, of rice straw. And what are we going to do with that stuff? Can we turn it into energy, into fuel, into feed, or into fertilizers? And often we can't. So farmers find that difficult for reasons that I'll explain. So what they do is they burn it. And that produces a lot of pollution, produces a lot of health effects. It has negative effects on yield because it produces ozone as well, and it's no good for global warming. And it's a waste of a potentially useful resource. So why don't they use it for biofuels and for bioenergy? One of the reasons is rice, as we've said, is a silicon accumulator. And rice straw contains a lot of silicon. And that reduces the palatability for animal feed, and it reduces the use in bioenergy because it clogs up the boilers. So high silicon is a problem, and you can see this is the rice burning season. In the Punjab, you can see all this is a satellite image. All this is fires. And it's producing huge amounts of pollution that's blowing onto Delhi. In Vietnam, the problem is trying to cross the road when it's absolutely clogged with people producing pollution from these motorbikes. So what can we do? Well, we can improve the palatability of rice straw to the animals that are reared in this part of the world, water buffalo or caribou, not caribou, don't get confused, caribou. And we can try and generate some bioethanol from the rice straw to put into these 1,000,001 motorbikes. And here we are, all standing around admiring this bioethanol-fueled motorcycle. Those are my feet, by the way. Nice flip-flop, Sue. So this is an international, interdisciplinary program. And this delightful creature is Professor Simon McQueen Mason. And Simon is very interested in cell wall digestibility. He's worked for many years on this organism, a wood-boring eating crustacean called the gribble, which can digest cellulose, and he's interested in digestibility of cell walls. And I say ecologists don't look like they're study organisms. It would be most unfair, but I do think there's a similar sort of expression, I feel. Anyway, so here is the team. Here are the aim of the projects. Increase the understanding of the role of silicon, identify rice accessions with contrasting digestibility and silicon content, assess the impacts of increased digestibility on animal feed with feeding trials, and illustrate to farmers the benefits that they can get from doing something other than burning. So just a quick run through where we are. We need to link the phenotypic variation in silicon content with genotypic variation in the cultivars. So we have a diversity panel from Vietnam and one from the Philippines. They've either been genotyped or they have been genotyped, so they've been very extensively sequenced. This apparently means something to geneticists. There's one single nucleotide polymorphism per kilobase, so it's a very detailed genotype, basically, that's what that means. And we're using PXRF to rapidly assess the silicon in those, so that's kind of high throughput phenotyping. And then we can use genome-wide association scans, apparently, to identify quantitative trait loci for straw digestibility and silicon content. And that means we can generate markers for breeding to improve rice straw. I say we, but of course, this is my bit. So where are we with this? Well, interestingly, there's a large range in digestibility of rice straw from the Vietnamese panel. You can see these are the different lines, and you can see there are some that are very highly digestible and some that are very low digestibility. But interestingly, the relationship with lignin isn't that strong. What's stronger is the relationship with silicon. So this is the results from the Vietnam panel again, big range in silicon content. And that is quite well correlated with digestibility, measured as the release of sugars from the straw. So silicon is obviously having a big impact on the digestibility. Why might that be? Well, we've already identified one QTL. So these are the phenotypes, silicon content and sugar, and this genotype A has low sugar release and high silicon, and this genotype G has high sugar release and low silicon. And that QTL is on chromosome 6 and maps to a gene that is known to be important in rice in structuring the cell wall. It creates some of the crosslinks within the cell wall. So we're now very interested to see how silicon relates to that building of the cell wall in rice. So we've got some QTLs, we've got a strong relationship between silicon and digestibility, we've got a QTL. So more work on the candidate genes, then the feeding trials, and then the crossing into elite lines. So far so good, and that project is very much continuing. And I just want to end with a mention of how silicon, all singing, all dancing silicon, can not only help indirectly in climate change through digestibility changes in rice straw, but can actually directly influence CO2 levels in the atmosphere. And this is a project led out of Sheffield by David Beerling, the Leverhulme Centre for Climate Change Mitigation. And David's idea is that if we can increase silicon weathering, we can increase the drawdown of CO2 and its deposition into the oceans. And that will not only, in the short term at least, remove atmospheric CO2 from the atmosphere, but it will also reduce acidification of the oceans. Silicon really is the answer to absolutely everything. So the idea is to increase weathering by sprinkling basalt all over the place. You just grind it up and sprinkle it on. Now, you're probably thinking, what the hell? But let me convince you. Actually, David's looking slightly skeptical himself. This is a £10 million project. I think he's panicking he might have to give the money back. But Emma, our PhD student, with David, Yuri, and Ton, and myself, full of youthful optimism. And it turned out that Emma was right. Because silicon, if we have enhanced rock weathering, we might well affect climate change, but we may well have benefits of food security as well. So what Emma did was grow her maize plants with basalt, and amazingly, when you add basalt to the soil, you get more silicon in your maize. So it does work. And then you find that when you look at the impact then on this northern corn leaf blight, which is a serious disease problem for maize, she found that the basalt treated maize had lower hyphal length of the disease. So basalt not only was increasing silicon uptake, but it was improving disease resistance. So that serves us right for being skeptical, doesn't it? Okay, so I'm going to wrap up now and just highlight, well, I hope I have highlighted, the value of interdisciplinary research and international collaboration. And that's how we can generate real-world impact. I've moved from studying classic plant herbivores and natural ecosystems to rather different plant-herbivore interactions and agricultural ecosystems. But it's been hugely valuable. And I think we all know the problems we have to deal with. We need 50% more energy, 50% more food, 30% more water, and all against a background of climate change. But we can address these problems. We can fix them. And in agriculture, we've got fantastic examples of innovation. We've been innovating in agriculture for thousands of years. And we can innovate to address these problems. And I hope I've shown you that I believe that ecology is at the heart of that innovation, that it's ecologists that can generate the ideas and the solutions to these real-world problems. So let's get out there and get on with it. Thank you very much.